Welcome to this comprehensive overview of a theoretical 90-day biological age reversal framework. In this presentation, we will explore the science, the math, and the mechanisms of sequential cellular rejuvenation. We'll break down complex clinical concepts into accessible everyday language, giving you a clear understanding of how this multi-phase protocol is designed to work. What is biological aging? To understand age reversal, we first have to understand aging itself. Biological aging isn't just one problem. It's a composite of 9 simultaneous failures, including genomic instability, telomere attrition, and mitochondrial dysfunction. Think of your body as a city. Aging isn't just one building falling apart. It's the roads cracking, the power grid failing, the garbage trucks stopping, and the repair crews retiring all at once. Because these hallmarks interact synergistically, addressing just one yields limited results. The core principle, why sequence matters. The core principle of this framework is that regenerative compounds cannot be given simultaneously. Biological logic demands a rigid sequence. Clinically speaking, administering regenerative compounds into an environment full of senescent cells would stimulate damaged cells alongside healthy ones. To use a simple analogy, you wouldn't repaint a house that's full of termites. First, you exterminate in phase 1. Then, you repair the wood frame in phase 2. Finally, you repaint and furnish in phase 3. Phase 1. Overview. Senolytic clearance. Phase 1, which spans the first 14 days, focuses on eliminating senescent cells, often called zombie cells. These are cells that have suffered irreversible growth arrest, but refuse to undergo normal cell death or apoptosis. Instead, they secrete a toxic cocktail of inflammatory cytokines. It's like having employees who stop working but refuse to quit, spending all day complaining and making everyone around them sick. Phase 1 uses compounds like FOXO4-DRI and dasatinib to effectively fire these zombie cells. The FOXO4-DRI mechanism. Let's look closer at how FOXO4-DRI works. Senescent cells evade death by using a protein called FOXO4 to lock away p53, the cell's self-destruct button. FOXO4-DRI acts as a decoy, competitively disrupting this bond. Once freed, p53 translocates to the mitochondria, triggering a Caspase cascade that selectively kills the senescent cell. In simple terms, FOXO4 is a bodyguard locking away the self-destruct button. FOXO4-DRI distracts the bodyguard, allowing the button to be pressed. Healthy cells don't need this bodyguard, so they remain completely unaffected. The SASP cascade. Why is clearing these cells so critical? Because of the senescence-associated secretory phenotype, or SASP. Senescent cells secrete inflammatory factors like IL-6 and TNF-alpha, which induce senescence in neighboring healthy cells. It's an exponential inflammatory cascade. Mathematically, the systemic burden is the sum of the secretory output multiplied by the number of senescent cells. Like one rotten apple spoiling the barrel, each zombie cell sprays toxic aging juice on its neighbors. Removing the source stops this chain reaction entirely. Rapamycin and mTOR, the autophagy switch. Once the zombie cells are eliminated, the debris must be cleared. This is where rapamycin comes in. Rapamycin inhibits the mTORC1 complex, which flips the master switch in your cells from growth mode to cleanup mode. A process called autophagy is the equivalent of calling in a professional cleaning crew to remove all the debris left behind. By reducing mTOR signaling by roughly 80%, autophagic flux increases 3 to 5 times above baseline, systematically clearing damaged organelles and protein aggregates. Phase 2. Overview. Epigenetic reset. Moving into Phase 2, from days 15 to 45, the focus shifts to reprogramming your DNA and recharging your cellular batteries. With the toxic burden cleared, we can target epigenetic drift, telomere attrition, and mitochondrial dysfunction in the remaining healthy cells. If Phase 1 was about clearing the junk, Phase 2 is like reformatting your computer's hard drive back to factory settings and replacing the old battery with a brand new one. Epitalon, turning on telomerase. A key component of Phase 2 is epitalon. Your chromosomes have protective caps called telomeres, much like the plastic tips on shoelaces. Every time a cell divides, these caps get shorter, Epidolon acts as an epigenetic switch, uncoiling age-silenced DNA to turn on the HTERT gene. This activates the telomerase enzyme, which physically adds new DNA repeats to the ends of your chromosomes, essentially resetting the cell's division counter and extending telomere length. The mathematics of telomere attrition. We can model telomere dynamics mathematically. Normal telomeres shorten at a rate of 50 to 100 base pairs per year, eventually hitting a critical threshold called the Hayflick limit, where the cell becomes senescent. Imagine your telomeres as a burning candle. Epidolon intervention is like adding wax back to the candle. As long as the rate of telomerase activation exceeds the annual attrition rate, the telomere actually grows, stabilizing the cell and reversing this specific metric of aging. SS31, repairing the mitochondrial power grid. While Epidolon works on the DNA, the peptide SS31 repairs the mitochondria, your cellular power plants. Aging damages the inner mitochondrial membrane, causing it to leak reactive oxygen species, or toxic sparks. SS31 is a positively charged molecule that binds to cardiolipin on the inner membrane. It acts as a repair patch, stabilizing the electron transport chain and reducing toxic leakage by 40 to 60%. The result is clean, highly efficient cellular energy production. NAD+, the cellular fuel. None of these repair processes can happen without fuel, and that fuel is NAD+. Unfortunately, NAD+ declines exponentially with age, dropping to about 50% of youthful levels by age 50. It's the gasoline that runs your cellular repair machinery, including sirtuins and PARPs. Phase 2 utilizes NAD+ precursors to refill the tank, ensuring that all the newly activated repair enzymes have the energy they need to execute their functions. Phase 3, overview, tissue remodeling. Finally, we enter Phase 3, from days 46 to 90. With a clean cellular environment and restored energy, the body is ready for tissue remodeling. This phase targets the extracellular matrix, vascular architecture, and hormone pulsatility. Think of it as the final home renovation. New paint in the form of collagen, new plumbing in the form of blood vessels, and turning the thermostat back to young by optimizing hormones. GHK-CU, the master gene expression reset. A major driver of Phase 3 is GHK-CU. If Epidolon opened the DNA instruction manual, GHK-CU is the foreman who reads it and tells the workers what to build. It acts as a master dimmer switch, modulating over 4,000 human genes. It turns up youth genes, increasing collagen synthesis by up to 70%, while simultaneously turning down aging genes associated with inflammation and tissue breakdown. Growth hormone restoration Phase 3 also focuses on endocrine restoration. Youthful growth hormone is released in sharp pulses, like a heartbeat for regeneration. With age, these pulses flatten out. The combination of CJC1295 and ipamorelin acts synergistically on the pituitary gland. Like two keys turning simultaneously, they restart the youthful high-amplitude pulse pattern, particularly during deep sleep, driving systemic anabolic repair. The complete cascade. When we look at the complete cascade, we see compounding returns at each stage. It's a relay race. Runner 1, senolysis, clears the track by removing the toxic zombie cells. Runner 2, reprogramming, sets the pace by resetting epigenetic marks and restoring energy. Runner 3, resurgence, sprints to the finish by rebuilding tissues and hormones. Skipping any phase compromises the entire protocol. Theoretical outcomes. What are the theoretical outcomes of this protocol? Extrapolating from preclinical data and isolated case studies, we see profound potential. This includes a 33% increase in telomere length, a massive reduction in senescent cell burden, and restored tissue homeostasis. If everything worked perfectly, your cells would behave like those of a much younger adult, effectively taking 15 to 20 years off your biological clock. The Horvath epigenetic clock. We measure this biological age using the Horvath epigenetic clock. Scientists can read your DNA by checking 353 specific spots for chemical tags called methyl groups. The pattern of these tags reveals your true biological age, which can be very different from your chronological age. The ultimate goal of this framework is to shift those methylation tags back to a youthful profile. Limitations in epistemic honesty. However, epistemic honesty requires us to acknowledge the limitations. This is not a time machine. While you can rejuvenate cells, you cannot reverse macroscopic structural degradation. Calcified arteries, permanent neuronal loss in the brain, and structural cartilage destruction in osteoarthritis cannot be fully undone. It's like putting a brand new engine in an old car. It runs beautifully, but the dents in the frame remain. Conclusion and key takeaways. In conclusion, sequential cellular rejuvenation represents the absolute frontier of longevity science. The logic is clear. Clear the zombies, reset the code, and rebuild the body. While it carries the theoretical potential for 15 to 20 years of biological age regression, it remains a research framework, not medical advice. Thank you for joining this exploration of the future of human health span.